is a single clone that it's really dominant and that has been selected among the other tumor clones. So we can see the difference between normal B-cell repertoire, and then after oncogenic events, we get to that monoclonal profile or state. The importance of sequencing the B-cell receptor, it's because in 1999, two works describing the different outcome in patients which had mutated or non-mutated IgHV sequences, and they proved that they had different survival rates. So in the case of the patients who shared more than 98% of their sequence with germline, they had an inferior survival rate compared to those that had more mutations. And so they were divided into groups known as mutated and non-mutated. So when TP53 gene is not mutated, the next step as an important prognostic biomarker is to do the mutational status of the variable region of the heavy chain locus. It's also known that there's a biased usage of the immunoglobulin VDJ genes, and also what is known as a serotype BCRs that are common to be combinations of different B, J genes, and also D and CDR3 motifs of sequences that are clustered into groups for their prognosis. So there is an European research initiative on CLL called airr-seq, whose last updated guidelines to assess the mutational status of the variable region of the heavy chain locus were proposed in 2017, and they include the following steps, which are the amplification of the region using leader primers in multiplex, including all the B subtypes. And in the cases we see in this image where we can find two DNA bands in a patient, then the separate PCR for all the subtypes is done, so we can detect the different subtypes that are present. Then these are sequenced using Sanger, and the analysis method that is established is to use the International Immunogenetics Information System tool, which is the IMGT Quest, which assigns the VDJ genes, CDR3, and then they can be classified into non-mutated and mutated CLL clones. To do this report of the mutational status, we have to be very careful with patients in the boundaries between mutated and non-mutated, as they can have variable behaviors, not necessarily included into their groups. And also after that, we can classify them using subsets that are already established to have a prognostic value, such as what are defined as the subsets. They are numbered. So some of them are the 1, 2, 4, 8. And for example, it's really interesting to note the group 2, because it only implies the presence of the B gene 321, and it's always a bad prognostic signal. So the limitation of this protocol I have shown is that we cannot detect that those clones that are minoritary respecting to that dominant clone. And it has also been suggested that subclones may also have an important role in the outcome of CLL. And the ideal thing would be to do high throughput sequencing to detect not only the predominant, but also secondary clones. But since we have more data than we can manage, it's not yet standardized. So what we are doing currently is to try to find the implication of these subclones, study them using HTS, and find what could be the improvement in the clinical practice when we include this subclone characterization. So we sequenced DNA from CLL patients using an in-house method that combines 3 different primer sets from the Biomed 2 consortium, which are all the framework region primer sets, framework 1, 2, and 3. And we have done this because we wanted to sequence them using shorter read sequencing, 150 bases per side using Illumina to improve read quality, and then obtain a whole coverage of the region with different amplicon lengths to overcome the fact that they can be mutated, and some of the sometimes we may not detect those alleles because of the mutation. So we have different possibilities to amplify them. And also it's an advantage for paraffin samples because DNA is fragmented, and having different sizes amplicons can help to these samples to be amplified. So for this purpose, we created a custom PRESTO pipeline. First up, for the preprocessing, including steps for quality filtering and a few steps for moving the primers, because we have different sets of primers and different orientations, and always annotating which is the primer that is removed, so we can know in both read 1 and 2 which is the B subtype that we are having on each sequence. And after collapsing the sequences, we do different strategies for the assembly. And the first one is for the amplicons like framework 1 that do not overlap read 1 and read 2. And so we need to do an assembly oriented by reference of the alleles. And then in the other cases, we do an assembly of the pairs, basically. And thanks to Susanna and Steve for this part. The next step was to define a threshold for the sequence distance to be considered when clustering sequence in the same clone, so to detect that ones that are clonally related. So as we can see here, we obtain a profile that it's different to other determinations, because it's we can see that it's really clonal in here, and we have a lot of sequences that share a lot of sequence, a lot of identity, and this threshold is what we define to differ between the what is clonally related and what is not clonally related. So following this, what we did was to pick sequences from 195 individuals from both Sanger sequences and the HTS sequences we had. And after annotating with IgBlast, we used this 0.1 threshold to define the clones. And we expected the Sanger sequences to be grouped inside the HTS clone that is more represented in means of reads, of sequencing reads. And what we first saw is that the mean number of clones, considering all the samples, is of 1.66. And in 97 of the samples, we detect more than one clone, about a 5%, it means of clonal percent. And also that the mean clonal percent for the dominant clones in HTS is the 76%. Also, we wanted to check if these Sanger sequences were grouped inside, mostly inside the major HTS clone, and we find that in 149 out of 195 subjects, we do detect that in the major clone. But for example, in 16 cases, we could not find the Sanger sequence in the HTS data. Afterwards, we wanted to compare if the mutation frequencies of both techniques could give the same information in means of determining the mutational status. And we compared the mutation frequency of the Sanger sequence with the median mutation frequencies of the HTS dominant clones, and we find that it correlates really well. So Sanger would be a good estimator of the mutation frequency of the whole clone. And just to show a result that is really promising, is that if we see this kind of representation for each sample in which we have the different clones from less represented to more represented, in this case, we have a borderline sample. because as we can see, this red dot here is Sanger sequence, and it's a borderline case because it has a mutational frequency that it's in the limit between the mutated and unmutated group, which is defined by this blue line here. And what we see is that we have both more mutated and less mutated sequences in both sides in the same clone. So we are only seeing one a little part of it when we characterize only with the Sanger sequence. And also what we observe is that when we have unmutated patients, so the Sanger sequence will fall here at the level of mutation 0, we still have mutation sequences with higher mutation degrees in that clone. So that's interesting to start studying. And well, this is what I wanted to present, and the conclusions are that the detection of Sanger sequences to start with this new analysis method in HTS clones was successful, 179 out of 195, and 83% of that successful detections, they were grouped within the dominant HTS clone. And also that Sanger sequencing is a good estimate for the moment of that median mutation frequency that we find in the clone, and that also it has a wide range of mutation frequencies. And we also want to study that 16 cases where we do not detect the Sanger sequence belonging to any other to any HTS clone. So to finish, I will like to thank my supervisor Javier Chavez of the Unit of Genomics and Diabetes, and the rest of my colleagues, and also my supervisor in the Unit of Hematology, which is Blanca Navarro, and my coworker Alicia Serrano, and the Client Science Lab, Steve and Susanna, and also the rest of the group. So thanks to all of you.